Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to query NCBI database programmatically. As a biologist, you must be extremely familiar with this database already. NCBI database is the world's largest repository for biological information. It is composed of over 40 specialized databases and hosts pretty much any biological information you can imagine. Searching on NCBI is straightforward. Just go to their website and search your keyword in their search bar. The website will then return anything that related to your keyword. Neat, right? But also problematic. Too much irrelevant information. That's when a programmatic search is needed. With the proper programs, we can specify exactly what we want to search, what format the output will be, and what filtering criteria we want to apply. One of such tools is provided by NCBI itself. It's called eUtilities. It's a command line tool which grants you enormous flexibility. Here is how to install it. On a Linux system or on your MacBook, open your terminal, go to the home directory, download the tool directly from NCBI, unzip it, add the path of the directory to your system. Basically, you just add this line of code to the .bashrc file on the Linux system or the .zshrc file on your MacBook. Go to the directory, copy and paste this code to the terminal and run it on your MacBook. If it's a Linux system, copy and paste this code and run it. Once the tool is installed, you can check the version by running the following commands. These are the core functions included in the package. I can't show you all of them, but I will show you how to use eSearch in the next few slides. Let me show you how to search a publication on NCBI using the eSearch command. For example, we want to search all the publications about COVID-19 published in 2024. We'll use eSearch-db means database, PubMed, query, COVID-19 in the title of the article, and published in 2024. pdate means publication date. This command will return metadata in the XML format. In this format, each piece of information is surrounded by a pair of tags. For example, our query string is surrounded by the query tag. The context shows you how many publications the search has found. Here we found 29,000 publications. Next, let's retrieve the complete publication list. Same command followed by the pipe. Hopefully, you still remember the function of the pipe. That means it takes output of the previous function as input of the following function. Here, eFetch takes the metadata from eSearch and returns a complete list of publication in the XML format. And then extract takes XML file and returns a human-readable publication list, and save this list into the PubMed search result.txt file. And here is the content of this file. It has the PubMed ID and the complete title of the publication. Similarly, you can search gene information or gene sequences using eSearch in the same way. Here, we search insulin in human. Database change it to nucleotide. Pipe the result to eFetch format faster and save the sequence in the FAST file. You can also save the sequence in the GenBank format by specifying format GB. This is what a GenBank format looks like. Searching gene information is done in the same way, except we change the database into gene. The results will be saved into a XML file. Of course, you can convert this format into a text file using extract for better reading experience. You can summarize the results using eSummary command, or specify the format using docsum format. All the results will be saved in an XML file, and you can convert them into text file if you want. The same tasks can also be performed in R, and it's even better. because the search results can be directly used for the subsequent statistical analysis or plotting. Here is how we do it. Open your RStudio on your laptop, install all the essential tools. The major packages we need here are rentrez and biostrains. rentrez for search and biostrains for sequence manipulation. Here is an example of searching publications using the rentrez package. Say we want to search all the publications about CRISPR between 2010 to 2024. The major function we use is rentrez_search. Database PubMed, term, CRISPR in the title, and the publication year. We do this search for each year in a loop. As_apply is a vectorized loop. The results contain a lot of information, but here we only need the count of the publication for each year. And this result can be directly plotted using the function plot. Type B means we want the line and the dots. You can search gene sequences or gene information in the same way. Basically, we use rentrez search to get the meta information and rentrez fetch to retrieve the complete information. You just need to specify the database, search term, and how many entries do you want to get. You can also specify the format you want to save. Function rentrez search, rentrez fetch, and rentrez summary are the exact counterparts of eSearch, eFetch, and eSummary in the eUtility package. Best of all, you can get gene expression data directly from GEO using the GEOquery package. The main function of this package is getGEO. Input is the GSE series number. This function returns a list of only one element, which is the gene expression set object. This is how we get the object from the list. Using the function exprs to get the gene expression matrix, pdata to get the sample information table, fdata to get the gene information. Here's the catch. Most gene expression matrices are not in this format, so you might have to go to the GEO website and download the gene expression matrix manually. If you want to do some BLAST analysis, which is to compare your DNA or protein sequences against the ones in the database, here is how we do it locally. On a powerful Linux system, create a conda environment for BLAST. Install BLAST using conda. Download the official BLAST database update script. Make it executable. You can show all the available databases using the update script. Download your desired databases. Here, we use Human Genome and SwissProt. Make the databases available system-wide. For BLAST-N analysis, the input is a FASTA file which contains all your DNA sequences. Database is the human genome we just downloaded. Output is a text file. Out format is a standard format, just copy this one. We only need 10 results. It's the p-value 10 to the minus 5. For BLAST-P, you need a FASTA file that contains all your protein sequences. Database is a SwissProt database we just downloaded, and all other things are same. This is the format of the BLAST-N results, and this is the format of the BLAST-P results. This is all I have for today's tutorial. Subscribe if you like it, and I will see you in the next video.